everyone. We're joined today with Carly, who is a PhD student from the University of Reading, and she's going to share with us some information about herself and about her research. So tell us who you are, Carly.
Hi, I'm Carly. I am a PhD student at the University of Reading, and my PhD project is all about fusogenic liposomes and how we can use these liposomes in platelet research.
Cool. Well, that sounds really interesting. So when you get to the lab in the morning, what's the first thing that you turn on?
So when I get in the lab in the morning, it really depends on the experiments I'm going to be doing. But more often than not, I use something called a flow cytometer. So with this, we're looking at single cells, and we specifically want to look at markers which are on the surface of the platelet. Or I will be using the microscopes. So these are really important because platelets are so tiny, and again, we can look at fluorescent markers on the platelet.
So tell us a little bit more about your research.
Yeah, so at the moment, platelet research is highly reliant on animal models. And because we use a lot of mouse platelets, these are very different to human platelets. So my research is trying to focus on introducing a different way to investigate platelets, but in human platelets. So the idea is to use little carrier molecules or carrier vesicles called fusogenic liposomes. The aim will be to fill these little liposomes with cargo, specific cargo that we're interested in, and then hopefully use those to fuse with the membrane of the platelet and deliver that cargo to the platelet cytoplasm.
That sounds really interesting. So why do we want fusogenic liposomes to fuse with platelets? What's the overall aim of your project?
The overall aim would be to carry cargo into platelets to really try and identify different things within platelets, either different mechanisms, different pathways, so we can sort of interrogate that further, basically.
And so it sounds like a lot of effort as well to try and get these kind of cargoes into the platelet. Is there a reason that we can't do that already? Why is it so important that we learn how to do this?
Yeah, so platelets are really tiny and they don't have a cell nucleus. So conventional methods maybe that we'd use in different cell types or bigger cells just can't be applied to platelets. So this offers an alternative method, but it's very new. So yeah, it offers that other.
What inspired you to get involved with the project?
So I have worked in platelet research before I applied to do this PhD. So I was well aware of how platelets can cause problems within our bodies, and I wanted to continue that research. But this project offered something which I'd never really heard of before. I hadn't heard of fusogenic liposomes. The project seemed really, really interesting and exciting, and something different to what I worked on before, but still within the platelet field. So that's why I chose to do that.
Thinking about the big picture. So how might this research somewhere down the line kind of help people suffering from thrombosis or heart disease?
So at the moment, my project isn't really looking at anything like carrying drugs or anything like that into the platelets, or using markers to specifically go to the platelet. But fusogenic liposomes have been used, or liposomes have been used for drug delivery. At the moment, I think my project is still in the initial stages. So just to be able to go to get fusogenic liposomes to fuse successfully with platelets and use that as sort of more of an investigative tool as opposed to actually drug delivery. But fusogenic liposomes, as I said, have been used to deliver drugs, so there's no reason why that couldn't be an option maybe in the future. It just depends on how successful that is.
So it sounds like you'd be able to better understand, I guess, those pathways that are going wrong in heart disease, so then maybe kind of developing drug targets further down the line.
Yeah, absolutely. I think to understand different pathways of activation or different signaling molecules, and to be able to identify more novel sort of concepts, I guess.
An awful lot of scientific research is reliant on funding from charities or research councils, or funded by universities. Where does the funding for your project come from?
Yeah, so my research is funded by the NC3Rs, and they work really hard to reduce, refine, or replace the amount of animal models in research.
Great. So Carly, you're one of the committee members of the Platelet Society. What drew you to, or what made you want to take part in the committee and be part of the committee in the society?
A few reasons really why I put myself forward to be a committee member. First of all, I feel that it was a really great opportunity to be able to get involved more with the society and the charity as a whole, but also to be able to engage more with members and to be able to get to know people better that way. Also to be able to support ECRs, but being an ECR myself, I feel that I am always a really typical example of just having really bad imposter syndrome, and sometimes I find that really, really challenging for me and my work sometimes. So just to be able to give a bit of that kind of perspective as well. Just really the engagement as well. And also some of the things that I've been involved with, sort of organising meetings, organising events, socials, things like that. I've never really experienced that before, and it's all really good things to be involved with. That's a few reasons why I put myself forward.
Thanks, Carly. That was really great.
For more information, please visit the Platelet Society website at plateletsociety.co.uk or follow us on Twitter.
Join us next time when we'll be talking to someone else about the exciting platelet research in their lab.